Hello, everyone. Welcome to the Burst fifth general meeting. I'm so glad you guys are here. We have a really amazing list of speakers for you guys today, so I'm really excited for that. So let's just go ahead and get started. We're only going to do a few announcements this time because we want to save more time for our speakers, but you guys will be able to read the rest of the announcements in the post-meeting email. So our member of the bye week for this week is Trent. Trent, are you here?
Yes, I am.
Awesome. Well, congratulations. You guys don't know Trent, he's a freshman Burst member, and he's also in the academic events committee. He's been helping us out on a lot of the Burst events. So congrats, Trent. Ruhi will be emailing you about your free t-shirt.
Thank you.
Just a reminder that a minimum of 16 points needs to be earned by the end of the semester to remain an active Burst member. And so if you meet the 1447 requirement, you're good to go. And if you're part of a research lab, you're welcome to fill out the form that's linked on the slide to earn 6 points towards research credit.
All right, so those are all of our announcements. Like I said, you can read the rest of them in the recap email. So now I'm going to go ahead and introduce our guest speakers. We have three. First up is Dr. Scullin. Dr. Scullin, are you on the call?
Yep, I'm right here.
Awesome. Well, let me go ahead and make you co-host so that you're able to share your screen, and then I will introduce you. One sec. All right, so all three of the guest speakers that we have tonight all received the Faculty Early Career Development Award from the National Science Foundation. And this is a really prestigious honor from the NSF, and it's provided to young researchers, and it gives them significant funding to advance their research. It's a highly competitive award given to those with potential to serve as academic role models in research and education and to lead advances in their field. So last spring, Baylor earned a record number of 4 NSF Career Awards, and today we'll be highlighting 3 of those Baylor recipients. So first up, we have Dr. Michael Scullin. He is an assistant professor of psychology and neuroscience. He is also the director of the Sleep Neuroscience and Cognition Lab. And of course, he received the NSF Career Award to study the impact of sleep habits on a student's ability to learn complex STEM materials. So without further ado, Dr. Scullin, take it away.
All right, thanks for having me. I've been involved with Burst activities ever since I got to Baylor, and I think it's a great organization, and I'm always happy to share what I know about sleep and neuroscience with y'all. So my work is really the work of lots of people. And I just want to start off by saying that I'm blessed to have some of the best team, including undergraduate RAs at Baylor. I think if you're interested in career in science, Burst is a great place to start. And then once you get into sophomore year, do your best to get into a lab. Do your best to get into a lab that speaks to you, that you think you can be passionate about. And whatever lab you land in, make sure that you jump in with both feet. Treat it as your number one priority. That's the best way to get the most out of it, and really learn if research, if science is right for you. So we are interested in sleep from a couple of different perspectives. The first is the basic thing that your mom has been telling you for like 20 years now, which is sleep helps restore your body. That's absolutely true. It's true from a few different perspectives. One is we know you get sicker if you haven't been sleeping well. In fact, we know that you're actually more susceptible to respiratory infections, things like catching the cold. You are 4 to 5 times more likely to catch if you've been cutting back on sleep to 5 hours a night. Funny thing is, actually it's not funny at all, but it's sort of fascinating. If you've been cutting back on sleep and then you go get the vaccine for the flu, your antibodies, you have half the level of antibodies as you would have if you had gotten a full night's rest. So with the COVID vaccine, hopefully around the corner, fingers crossed, months away or whatever it is, make sure you're sleeping well beforehand. You want it to take its effect. We also know that losing out on sleep, you're just not going to perform your best, whether your sport is basketball or golf, um, tennis, football. All of these things are empirically validated that cutting back on sleep hurts your performance, getting more sleep benefits you. Um, and there's also a little something to the concept of beauty sleep. If you've been cutting back on sleep, and if you take a photo of yourself after cutting back on sleep and then a photo of yourself after a good night of rest, and then they're rated by others at like a different university, this paper has shown that you're rated as significantly more attractive, physically attractive, if you have been sleeping well versus if you've been cutting back. Mom was right. But that's not what my lab is interested in. We're interested in predominantly sleep's effects on the brain. And what I've done here is highlight all the regions in the brain that are significantly impaired if you either pull an all-nighter or if you just gradually cut back on sleep over time. And these regions are things that are very important to your ability to reason. So reason out like test answers, but also reason morally. Like moral decision making actually changes if you've been cutting back on sleep than if you've been sleeping well. These are also regions that are very important to stress and emotion regulation. Life just feels worse if you've been cutting back on sleep. You're more likely to come to tears or to be driven to anger than if you've been well rested. And certainly, we've heard this for a long time, but it's absolutely true that sleep is really important to your ability to learn concepts in the classroom and to retain them over time. We've been studying Baylor students for a little while, and I don't know if you remember, but when you were asked to do that new to BU survey, whether it was this year or in previous years, there was probably an item there on how much sleep you're getting. And what we've done is we've learned that Baylor students, only about half of them are getting the amount of sleep that they should, the consensus guidelines that are dictated by medical and scientific societies, on how much sleep you should be getting. And it matters. This one little question on how much sleep you're getting in your first month of college is predictive of how well you're going to do in your first semester at Baylor for GPA, in your second semester at at Baylor for GPA, in your third semester, and in your fourth semester. And it's probably predictive beyond that. We just haven't gotten to that point yet in our data set. There's also kind of a weird thing about long sleepers. If you're routinely sleeping 10 hours, 11 hours, or 12 hours, that's normally a bad sign. It's not necessarily that sleep is hurting you. It's that you are fatigued so much that you have to sleep a lot. And if you're fatigued a whole lot, it's probably not very easy to concentrate in class. Not only does sleep and just self-reported sleep, like this is a really coarse measure, predict how well you're going to do, but it also predicts who's going to land on academic probation. So short sleep doubles your risk for landing on academic probation first couple of years. So what's going on? Well, we know, or we think, that when you're cutting back on sleep, it has an effect on the ability of your prefrontal cortex to concentrate, to think analytically, and to encode information deeply. And when those things aren't going well, you're more reactive to stressors. You get to see that test and just feel more anxious about it. And all of that feeds back into, I'm not going to learn very well, and I'm not going to show what I learned on my test very well, which makes you more worried, keeps you up at night, causes you to stop persisting as much, and feedback into the cycle. When we're not shut down by a pandemic, Our main line of work is in a sleep laboratory where we use something called polysomnography. We put electrodes all across the scalp and we measure brain waves while you are sleeping. Actually, people sleep better in our lab than they do at home. People average 8 hours and 15 minutes in our lab, whereas at home it's a little under 7 hours. What's cool about this technology is you can tell literally the moment that someone falls asleep. It's just, even just visually, just looking at the brain waves, it's very clear the moment someone is still awake and then they nod off. We can also tell when someone is in rapid eye movement sleep, which is when you're dreaming, eyes begin to move rapidly underneath the eyelids and your muscle tone is basically a flat line, you're basically paralyzed. This is why some of you have woken up and you haven't been able to move when you're awake. And some of you have woken up, not been able to move, and you like have some weird vision or you hear some weird auditory sensation and you kind of think you're going insane. No, it's just your REM sleep has protruded into your wakefulness. Probably not harmful, usually means you haven't been getting enough sleep. And we also can look at slow wave sleep, which is the deepest stage of sleep, and also the stage of sleep where it seems like the parts of your brain that are involved in memory processing begin to dialogue with one another. Other times they're quiet, slow wave sleep, everything else quiets down, the memory regions dialogue. I just want to leave you with this idea that some of us come in feeling like, I know sleep is important, but there's absolutely nothing I can do about it. I just can't sleep. For a number of years, we've done something called the 8-hour sleep challenge with students, and we've done it actually during finals week. Because during finals week, people sleep very poorly. If you tell people about how important sleep is, it doesn't really change much. They just continue to cut back on sleep to around 6 hours a night. But if I were to offer you extra credit for proving that you're sleeping 8 hours a night or more, then what we end up finding is we basically eliminate short sleep during finals week, and we get almost half of students to become 8-hour sleepers. And if we track how well students are doing on their final exam, so here's multiple choice, which are always easier than short answer. Short answer is more dependent on the prefrontal cortex. What we find is that with sleeping more during finals week, you improve your final exam score a little bit with multiple choice and a lot on the short answer questions. So that's the overview of our research, of our research program. If you're interested in being a participant in one of our studies, we're not doing the in-lab stuff just now, but we do have some other studies going on where you can actually be compensated $50 for participating. So if you're interested, you can email that email address, BaylorSleepLab@gmail.com, or you can just grab your phone, scan the QR code, and it'll take you to a screening page. And that's all I have. I don't know if I used up all my time or too much time. How did it turn out?
Yes, we still have some time left. So does anyone have any questions for Dr. Scullin?
Yeah, why don't we go through questions now? I know there's something at the end, but I've got little kids I've got to tuck in.
Hey, Dr. Scullin, I have a question. Are you able to hear me?
Yeah.
All right.
Hi.
So are these studies mostly correlational, or have you all had any that are more like you can prove causation?
Oh my gosh, I'm all about experimentation. Yeah, end this shutdown and we're back in lab and we're running our experiments. One of our big sources of funding right now is to bring people into lab, and then they're randomly assigned to sleep normally 8 hours a night for 3 nights in a row, or to cut back on sleep 5 hours a night. And we've got them in our environment, we've got them attached to our equipment, we've got really precise control over how much sleep they're getting. And then at the end of that week, we take them over to an MRI, fMRI machine, and we have them try to solve creativity tasks and learning tasks, either after being well rested or after being sleep restricted. And so we're going to see, how does the brain try to think creatively? How does the brain try to learn differently when you're sleep deprived versus when you're well rested?
Awesome. Very cool. Thank you very much.
I had a question. It was a kind of weird question, but so did you do a study like did you compare like test scores or GPA between students that kind of chose to sleep late and wake up early versus the students who were forced to do so? Like for me, sometimes I sleep late and I wake early to watch a sport or something, whereas some days I'm sleeping late and then I wake up early because I have a class. So is there a distinction between that?
Yeah, if I understand correctly, it's kind of like asking, is averaging 7 hours the same no matter how you get it, broken into nighttime, a nap, or broken into what time you're sleeping? And the answer is actually no, it's not the same, because the type of sleep you get, the quality of sleep you get, is dictated not only by how long it's been since you slept, but also by a number of circadian rhythms. So if you're varying what time you're going to bed and what time you're waking up, and it kind of yo-yos across the nights, it doesn't actually equal out to the same thing. So what we've learned, and what other labs have learned too, is that consistency of that bedtime wake time schedule is really important. It's important because it affects your sleep quality, but also it allows your circadian rhythms to align. So just think of it from this perspective. What if you were hopping on an airplane to go to London? You're going to feel jet lagged and you're not going to be at your best. And either you stay on that for a long time and you're okay, or if you get back on a plane and you come back over to the States, then you're going to be jet lagged the other way. And if you keep on doing that, it doesn't matter if you're sleeping 7 hours, you're always out of sync, out of alignment with the 24-hour cycle.
Thank you, Dr. Scullin.
I think we have time for one more question. There's actually one in the chat, Dr. Scullin, if you see it.
Have any of your experiments dealt with power naps? What's a power nap? What is the operational definition of a power nap? 5 minutes, 10 minutes, 20 minutes, hour and a half, 4 hours? Anyone took a nap that lasted 8 hours? I'm going to guess that the person means like a 20-minute nap. We haven't studied that before. We've studied 90-minute naps. 20-minute naps are fine. They give you the subjective impression that you're well rested. It has some benefits, but it's really better to just get good consolidated nighttime sleep. The more you fragment your sleep across a 24-hour day, the less likely you are to get quality, restorative, cognitively enhancing sleep at any time point.
Awesome. Well, thank you so much for being here, Dr. Scullin. We really appreciate your time.
All right, and if any of y'all have any sleep questions later, feel free to send me an email.
All right. So our next NSF award winning speaker is Dr. Ling. Dr. Stanley Ling is an assistant professor of mechanical engineering here at Baylor. He's also the director of the computational thermofluid lab. And of course, Dr. Ling received the Career Award from the NSF to support his research on spray formation modeling and simulation. So Dr. Ling, over to you.
Okay, can you hear me?
Yes.
Can everyone hear me? Okay, so I'm going to share a slides. So hopefully you can see properly. Can you see my slides, the presentation?
Yes.
Okay, wonderful. Yes, I want to introduce myself to you. My name is Stanley. I'm from the Mechanical Engineering Department. And today I'm going to introduce my research to you on multi-phase flows. Um, so this is my group. So a little bit about myself, also my group at Baylor. So I got my PhD degree from University of Florida, and then I stayed in the same group for postdoc for 2 years, and then I moved to Paris in France for my postdoc for a few years before eventually in 2017 I came to join Baylor. And I'm very blessed to have a very smart and diligent PhD students working in my group. So our research is not just myself, it's also a lot it contributed actually by the PhD students in the group. So, and this figure on the right, you can see here, is my overall research philosophy in a high level language. So we start from the green color, that we develop numerical methods and we develop simulation framework, and that's a fancy word to say software. So we develop software so that we can simulate the fluid flows, and then we use to study. We spin to the blue color, that's the fundamental multi-phase flow physics. We want to use the tool we developed to study the physics. And then after we improve our understanding about the physics, we use the knowledge to develop new techniques, multi-phase flow control technique. How can we control the flow better, so make all the engineering application involve multi-phase flow perform better. And then you can see this red color also spin continue back to the green color. Why? Because when you want to innovate something and want to study something new, you find out that, oh, my research tool is not sufficient. So we need to develop new methods and study new phases and then kind to find something new and improve the performance. So this is really like a iterative process. That's why I use this kind of wheel circle to represent that. And also in my presentation, if you want to have a question anytime, you can feel free to interrupt and jump in. So my research do a lot about spray and droplets. I don't think anyone here will feel, oh, what is a spray? Everybody knows a spray every day. I take a very simple example here, like a perfume. Okay, we see a lot of female students here. I mean, you use perfume. How do you use it? Do you pour it on your skin? No, you don't do it. It's very expensive, right? So you're not going to do that because for expensive liquid, the best way to use it is quite often is to have something like the cap of your perfume bottle to suck the liquid and then inject with a high speed and break into a small droplets. Okay, so they mix better, they evaporate better, and they deposit on your skin better. So it's a better way to use the liquid. Okay, and you may aware or you may not be aware that what happened on the perfume bottle is actually very similar to what happened in your car. Okay, and we all know that um, I mean, we want to have a car that is have a high mileage, right? It's a gas efficient, okay? And because the fuel itself is expensive, it's a important resource. If we can use it in a better way, it's good from the economics point of view and also for environment as well. So I saw this picture here. I believe everybody have seen a gas station like that. And you can see in this picture, you have a green nozzle and you have a red nozzle. Do you know what's the difference between them? This sounds like a silly question to ask. Okay.
One is diesel.
One is diesel, one is gasoline, right? Have you ever tried to put the diesel fuel in your gasoline car?
No, because that's how you destroy the engine.
No, I have done that by mistake. Okay. Actually my wife did it, but I did not stop her, so that's also my fault as well. So we have one time we have a gasoline car, but we put the diesel fuel into the engine. But the next question is, do you know what will happen to the diesel fuel? Can you start a car? Okay. So if you do the opposite, you have a diesel truck and you put gasoline in it, that will be more dangerous and more damaging to your engine. But if you put a diesel fuel in the gasoline engine, they will not burn at all because as the following, what I'm showing in this picture, all the liquid fuel we use in engines, they never burn in liquid. Okay, we always inject them, break them into very tiny small droplets, and they evaporate to vapor and mix with the air, and then it burn, and that would be an efficient way to burn, and you produce less pollution and so on and so forth. So diesel fuel is very hard to evaporate. So they need a special injector to break them into small droplets so that they can burn. And that's not in a gasoline engine. Okay, so you put diesel fuel in an engine, even you by mistake turn it on, but don't do it. Okay, to turn on your engine, they will not burn at all. Okay, because they cannot evaporate so fast. So at the end, you just have some liquid deposit into your engine. So this also, I mean, it's a funny example I make here, but this also motivates us to think about how we can inject the fuel into the engine, and that has a big impact to the performance of the engine. Okay. And if you have not seen an engine, open your car, open the trunk and have a look at it. It looks like big, but the injector is actually very small. Okay, so to be able to see what happened in this injection process is very difficult. Okay, and that's why in our lab and also some of my other colleagues, we developed a simulation tool to simulate the process so you can see the details. Okay, if I take a picture, you see what in the top row of picture, that's a spray. But in a simulation, in a good simulation, we're able to see how the liquid is injected and start to break and form small droplets. And we also can do quantitative measurement. How small is a droplet, where are they located? Okay. Not only the engine in your car, even the engine in an aircraft, it's a gas turbine engine. It's different type of injection technique, but they also need to inject the liquid fuel and break them into small droplets, vaporize before they can burn it efficiently. Okay, so on the simulation I'm showing right now on the right corner here, we have used more than 2,000 CPU cores in a supercomputer to simulate it so that we can capture all the details. Okay, so a very big overview about our research approaches is that first we need to have the physical model. We need to know what kind of equations you're going to solve. Okay, and they're difficult math equations. I always encourage students who are not very motivated to study math, and math is very important. All engineering problems eventually boil down to math problem. Okay, and because the equation is complicated, we cannot solve it on paper. Okay, so we have to write a program, computer program to solve these equations. Okay, so that's the next step. Okay, and we want to write a program that can not only can be run on a laptop. Okay, because use a laptop to simulate what I have just shown you, we take years and that's not useful. Okay, we need a supercomputer. Okay, so we need to develop a program that can be run in a supercomputer. Well, we use a supercomputer called Stampede 2 a lot in the Texas Advanced Computing Center in Austin. So they have more than 100,000 CPU cores. So if you are program, you have developed the tool can run well, they will give you computer time there, and that's how we do our research. So in my lab, it's very boring. If I invite you to visit my lab, you just see a bunch of computers over there. There's no lab, no equipment. But we use actually we use the supercomputer. That's our main research equipment. And after that, you run the simulation on those supercomputers. Sometimes we use 10,000 CPU and for weeks, and so that we can get the simulation is a very high quality as you see in this picture. So my as a I just been mentioned that I have a NSF career project to do spray modeling. So it's do the kind of work I just described to you. Okay, so specifically we look at a case that we have gas and liquid all come together when they inject it. So the high-speed gas and the low-speed liquid, they will kind of have a share on the interface and make it become unstable, forming the waves a little bit like what you see in ocean. When you go to the beach, see those ocean wave break. So they also break into droplets. So the bowl liquid will first break into a liquid sheet and then form ligaments and eventually droplets. And the grid you see here is something like a resolution of a picture. So if you have a good camera, have high resolution, high pixel, then you can see the picture very well. So it's the same way in the simulation. But not everybody afford to run a simulation like us using thousands of CPU. So this project is trying to use one very interesting idea. We want to develop a model, okay, that so that people can run the simulation with a coarser mesh, okay, less expensive simulation, but still capture the dominant thesis, okay. And the idea behind this project is based on a technique called skeletonization, okay. So on top of two simple example over there, you have a hand. Hand is a very complicated object, okay. But if you be able to subtract the skeleton of your hand, it's a much simpler representation of the topology of the object, okay. And the horse is similar. So we are trying to use this skeletonization to find a skeleton of the spray, okay. And based on that, we can develop the computer model so we can simulate it accurately but less expensive, okay. And this this model, this computer program we develop eventually is to help us to improve the injection technique. So for example, the left figure here is the convention air blast atomizer, one type of injector used in all the gas turbine engine. The question is that, can we improve it? Okay, if you have an idea, will you be able to test it before you really manufacture it and test it in an engine? Okay, and that's what we are doing exactly with the tool we develop in our research. So we are trying to investigate in a novel concept called flow blurring atomizer. So very similar operating conditions, we can generate a spray much more efficiently. Okay. And finally, I want to wrap up my presentation with the impact of our research. So our research goal, also including this NSF career project, is trying to establish a high fidelity simulation infrastructure that we can simulate a wide range of different spray application. Okay. One of them that we are interested in doing is related to the COVID that everybody is suffering right now. Okay. You may be aware or not, when you sneeze, you generate more than 80,000 jobless in one sneeze, like as shown in this picture. Some of them are big, some of them are small. Like in an engine, the liquid fuel, the gasoline, or diesel fuel injected into engines. Can we predict the size of the jobless? And can we predict how it's going to propagate in the air? Which jobless will fall on the ground? Which jobless they will suspend it in the air and evaporate and dry up to be an aerosol. So this all related to how we develop the guidelines for people to obey to prevent the disease propagation. And also our research, we have an educational purpose, is to educate the public about computer modeling using the supercomputers. That's what they call the high performance computing facility. We work closely with the Mayborn Museum to interact with them, to develop exhibit and also event to interact with the children. And we in particular passionate to interact with the children with physical limitations. Okay, we want to encourage them. Yes, I mean, you may have some physical limitation that you may not to do the engineering work 50 years ago. Okay, but now with the computer and modeling technique people develop, you actually can become an engineer. So that's what really the message we want to convey to them. And every time I get an opportunity, I will share this dream with my students. Okay, so I think that's what I want to talk. I would welcome any question if you have.
Dr. Ling, there is a question in the chat.
Okay.
Do you want me to read it?
I can exit and read it myself if I can. Okay, so I see a question. How do you measure efficiency of the sprays? Very good question. So you can think about in your engine. So a bad fuel spray or a bad injector, if your injector in your dish, if you happen to have a diesel truck, injector if it's broken, then with the same pressure driving the flow, okay, same pump, okay, then you cannot generate such a small jobless, okay, then it would be bad. And those big jobless will deposit on the wall of the engine and they do not burn efficiently. So that will not be an efficient way to generate a spray, okay? And also, I mean, if you have anything at your home, your perfume bottle, if it's stuck or something, and you push it, you generate the jobless kind of big, and also you realize that that's not efficient. Okay, the efficiency is decreasing. Okay, if a good technique, I mean, good injector, good design, then they can always generate the job list in the size they want, okay, and also spread them in the angle they want. And that's what people try to control in a lot of engine operation.
I have one question.
Yes, Elizabeth, please.
So if you find that one of these sprays is inefficient, how do you go about making it more efficient? What
Yeah.
kind of like, what's your thought process on going about that?
Yeah, so first of all, we need to think about, hey, what is the property of the liquid I want to generate spray? Okay, if you want to generate water spray or a diesel spray or a biodiesel spray nowadays, that the biofuel become very popular and they're different. And the biofuel are much more viscous, like honey. So they're more difficult to atomize, to generate spray. Then you need to change the operating condition of your nozzle, also can change the injector and also the geometry and the size, the design of the injector to make sure it can deliver what you want. Okay, does this answer your question?
Yes, thank you.
Thank you.
All right. Well, thank you so much, Dr. Ling, for being here. We really appreciate you talking about your research with us.
No problem. It's my great pleasure. If you have any question, I mean, contact me anytime. I love to interact with our students for the research opportunities. Okay, thank you.
Awesome. Thank you. All right. So our third NSF award winner is Dr. Alicia Gallagher. She is the Assistant Professor of Chemistry and Biochemistry here at Baylor. She is also the director of the Gallagher Group, which is a bioanalytical research lab that focuses on characterizing glycosylation. Dr. Gallagher received the Career Award to propel her research towards developing new methods for analyzing and understanding the function of glycans. So Dr. Gallagher, over to you.
Okay. Do we see the normal screen presentation?
Yes.
Okay. So thank you for inviting me. Today, my plan, since this is a shorter presentation, is to try to give more of a general overview about what we're doing. If there are more detailed questions, I'm happy to answer those. But with that, we'll go ahead and get started. So just to give you an overview of kind of my background, I went to the University of Arizona, and that's where I got both my bachelor's and my PhD degrees. My bachelor's, I have a double major in chemistry and molecular and cellular biology. My PhD is specifically in analytical chemistry, and this is actually what Arizona looks like. This was taken not far from my apartment while I was there. After that, I moved to Washington, D.C., where I worked in a government lab. And so I worked at the National Institute of Standards and Technology. And so this is the National Measurement Lab in the US. They do fall under the Department of Commerce. And so a lot of the projects that I worked on were relevant to characterizing pharmaceutical compounds. And so after my postdoc, I then moved to Baylor. And as Shubi mentioned, I'm a bioanalytical chemist. And so we're going to talk about what that actually means. I also enjoy musicals, so shout out to Hamilton. So with that, I know that this is not an in-person seminar, so things are a little weird on Zoom, but any chemistry or biochemistry majors out there? See a couple of hands. Okay. So for those of you that aren't in chemistry, we often describe chemistry as containing 5 different subfields. And so we have analytical chemistry, biochemistry, inorganic, organic, and physical chemistry. And so when most people think chemistry, they think about making molecules. And so this is particularly relevant for organic and inorganic chemistry, where we focus on making molecules with either carbon or often metals. And so that would be inorganic chemistry. Biochemistry is the study of biomolecules, so things like proteins, DNA, RNA, things like that. But my group is an analytical chemistry group. And so if we think about that, analytical chemistry, as defined by the American Chemical Society, is the science of obtaining, processing, and communicating information about the composition and structure of matter. In other words, we care about what something is and how much of it exists. So as an analytical chemist, we're really focused on how do we take measurements about chemicals or about molecules. And so one question could be, well, then how is analytical chemistry used? It's regularly used to look at safety and quality of both food and pharmaceuticals. It is used to ensure compliance with environmental regulations, so looking at contaminants in air, water, lab spaces that people work in, things like that. It's often used to support legal processes with forensic measurements. So these could be things like analysis of drugs. They could also be more complicated. Analytical chemistry is also used to help physicians diagnose disease, so determining if particular markers are present that would indicate that somebody has disease. And it can also be used to provide measurements for various trades and commerce activities. And so what should be apparent then is that analytical chemistry is highly related to a lot of other topics, whether that's, again, forensics, biology, material science, environmental science, medicine, etc. And so I think that this is one of the best things about analytical chemistry, is that it's so diverse in terms of the applications that can be applied. And so it was already mentioned that I'm a bioanalytical chemist. So what is that? So I know you guys, this is talk number 3. So this is your opportunity to answer a question. So if somebody could either type into the chat or shout out, but when I say sugar, what comes to mind? Anybody?
The chat is blowing up. I don't know if you can see it, Dr. Gallagher.
I can see it now. So sweet, glucose, fructose, sucrose, candy. I like that answer. So often when I ask this question, I get these types of answers. And Halloween wasn't that long ago, right? So you may have purchased some of these things. Maybe if you're thinking about it from a healthy perspective, you could think about other sugars like fructose or sugars in honey, lactose from milk. What about when I say carbohydrate? Bread, starch, pasta, potato. I haven't eaten dinner yet, so I'm getting hungry. And again, normally what we think about in society are things like bread, potatoes, corn, starch, etc. And this is one way that we think about it in terms of nutrition. But in reality, sugars and carbohydrates, when we talk about the chemicals, they have they mean the same thing. A carbohydrate is a historic term because it can be translated to a hydrate of carbon. And the first purified sugars, like glucose, had a formula of C6H2O6. And so they looked like they were this hydrate or water hydrated carbon. A more current definition is that we have a polyhydroxylated aldehyde or ketone. So that's a very chemically sounding word. But when we look at molecular structures here, where these lines come together, these represent carbons. And so a polyhydroxylated structure, well, a hydroxyl are these OH groups. So we see that there's lots of these in these structures. And an aldehyde or a ketone basically is where we see this carbon double bonded to an oxygen. And so this is going to be consistent for any carbohydrate or sugar that we talk about. Now, one of the really interesting things about carbohydrates is chemically they can form these ring structures. And these structures, particularly these two, may look very similar. But for the non-chemists in the group, one of the difference here is the position of this particular hydroxyl. This difference in this position can actually give these two sugars very different biological structure or biological functions. Additionally, some sugars can also form these 5-membered rings. which again can have these very small structural changes that then can have big biological differences. And some of those biological differences may just be our ability to digest the sugar. Some sugars we don't digest the same way, and therefore we get the sweet flavors without having the calorie content. Now, besides this energy and metabolism idea, sugars are also really relevant for various materials and products. And so this is actually my favorite example. So Febreze, what they tell you is that it actually, traps odorant molecules and it takes them away. And so if you look at the structure of Febreze at the chemical level, we see a lot of these rings again, and they have a lot of these hydroxyl groups present. So this is a ring of sugars, and this is accuracy in advertising, I guess. But the center of this molecule can actually trap other molecules that have a smell, and they can take them out of the atmosphere, so you no longer smell them. So these sugars as well have materials properties that make them useful too. Now, in particular, my group is really interested in the health implications of carbohydrates. And so, any of you know your blood groups? Anybody? Some not? Okay. Well, these blood groups actually describe sugar structures that are present on your blood cells. And so each of these different shapes in different colors is a different sugar, and they can be linked together. And so what we see here we have a different sugar here present if you have an A blood group versus a B blood group. And if you have this H antigen or an O blood group, you don't have a sugar there. And so these sugars decorate all of your blood cells, which is why we call them blood groups. But beyond that, they decorate all of your tissues. And so if anybody knows somebody who's had like a kidney transplant or a transplant of any sort, it's really important that they look at matching blood groups. Well, that's not just because of the blood, but because the sugar structures cover all of the tissues in your body. So other examples of glycans, if you have taken biology, we're commonly looking at cell membranes that look something like this, where you have this cartoon, you have this lipid bilayer, maybe you have some proteins in the bilayer. But the reality is that our cells are covered in these layers of sugar. And so here we can see an image where we have a cell. Here in the bottom, this is the plasma membrane. And at the top here, this is the layer of sugar-based structures at the cellular surface. And so these sugar-based structures are present on all mammalian cell surfaces, as well as in secreted proteins. And these sugars can affect things like how proteins fold, their stability. They also can affect how cells adhere or signaling between cells. And these structures are seen to change in a variety of different things, like in immune response and in other disease states, like cancer. And so these sugars are really important then in terms of biology. Now, in the last presentation, there was a shout out to SARS or COVID, and so this is my shout out to SARS. But this particular virus has a protein. And so here we can see the different domains of the protein. And we can also see all of these little Y-shaped structures are indicating where there are glycans present in this protein. When the protein folds, it has a shape. And this is the S protein that sticks in the membrane of the virus. And what you'll see here is all these colored portions are actually glycans, or these sugar-based structures that cover the protein part of the virus. Now, why does this matter? The glycans are flexible, so they can move around. And so when you look at how these glycans move around, you can see that they actually cover the majority of the protein. Well, a lot of antibodies have been developed to detect protein components. And so if your body is trying to identify or detect protein components, these glycans help prevent that from happening. And so this is actually a way that a lot of viruses or pathogens in general evade detection by the immune response within our own bodies. So these molecules can have significant implications in terms of the immune system as well. both in terms of molecules hiding or viruses hiding from the immune system, as well as interactions with the immune system. So when we look at these structures, they're fairly complex. So when we think about other biological molecules like proteins or RNA or DNA, these are linear polymers. So you have a single amino acid attached to the next amino acid, attached to the next amino acid, et cetera. Whereas in glycans, we can get these branch units that show up. And so here we can see, again, each shape and color represents a different monosaccharide, but we can get these highly branched structures. And just like we saw in the SARS spike protein, these glycans, when they're attached to proteins, they can actually take up a significant amount of space. And so they can affect what the body sees. Now, another thing to realize with glycans is that they're not part of the central dogma of biology, where we have DNA gets transcribed to RNA, RNA gets translated to protein. Really, these glycans get added when we have enzymes that attach them to proteins. And so there's nothing in the DNA specifically that says this sugar should be added to this position to give this molecule a certain structure or function. And so this actually adds a lot of heterogeneity to their structure. And there's some hypotheses about why this might be in terms of, again, invading immune response, things like that. Another thing is that these glycans get added in the endoplasmic reticulum and the Golgi apparatus, and this is a fairly complicated enzymatic process. And so it takes a lot of different enzymes to make certain structures. So the structures that are formed are really dependent on, number one, the sugars that are present in these different organelles, as well as the enzymes that happen to be present. And so the end result is that there's a lot of heterogeneity in these structures. If you translate the same protein 5 times, each one of those may have a different glycan attached. And that can then make it harder to characterize what's the biological significance of these molecules. Additionally, like we've already talked about, they're branched molecules, so that in itself makes them a little harder to characterize. And the positions of these hydroxyls, whether they're pointing down or pointing up, whether these sugars are connected at different positions, all of these things may appear to be small structural changes, but they have really significant biological effects. And so for a long time, these molecules, just because they're hard to characterize, have been ignored. People have just chopped them off their proteins and said, it's fine, it's good enough. But I think what we can see with the SARS example, for that spike protein with COVID, these can have a very significant biological impact. And so we really need tools to characterize them. And so when we think about what type of questions do we want to answer about carbohydrates, my group is interested in what's the chemical structure, what's the function, and then for biomolecules in general, we think about the structures correlated to function. So by better understanding the structure, we can learn about the biological function. So my group, being an analytical group, we're really invested in developing new methods to characterize carbohydrate structures. And so this is where we don't have time to go through everything, but my group does do a lot of mass spectrometry. And so these are images of 2 different instruments that we use. These instruments measure the mass to charge of these sugars. So by knowing the mass, we can know the chemical composition that makes up the sugars, and then we can use some fancy techniques that allow us to get more information about the structure, things like ion mobility. We do some other experiments as well where we pull these tiny glass tips so that we can spray our samples directly into the mass spec. One of the things, again, that we're interested in is kind of the biological implications of these glycans or these sugar structures. And another thing that they do, besides just covering proteins, is that they actually provide binding sites on our cellular surfaces. And so these binding sites can enable pathogens to bind and be internalized into cells, and therefore they can have whatever physiological properties that they would have. So this is an example with cholera toxin. So cholera toxin causes cholera. And this particular protein structure binds to sugars that are present in various cells. And so by binding, they can be internalized into the cells and they can initiate all the bad symptoms of cholera. Another example would be like the flu virus. So the flu actually has a protein that binds to glycans or sugars in your respiratory tract. And in doing so, it's internalized, and that's how you get sick with the flu. One final example would be, has anybody heard that there are problems with antibiotics, like they're not working like they should be? So one hypothesis right now is, again, a lot of bacteria bind in the same way to cells in our bodies, where they bind to these sugar-based structures. So potentially, if you could interfere with this binding interaction, you could minimize the need to have to kill the bacteria. You would just prevent them from binding, you'd prevent them from infecting our tissues. And so this is another area of interest for us. So we care about glycan structures, but we're also interested in their binding interactions and how they relate to function. One of the ways that we do this is with a technique called native mass spec, where basically we transition from a solution phase where our proteins are present. So in biology, they're an aqueous solvent. That's what our body is. But in mass spec, we analyze gas phase structures. And so we need a way to gently transfer proteins from solution into the gas phase. And when we do this, we think about, proteins want to stay in solution. So this has to be a very, very gentle way to get them into the gas phase. And so here, what we're looking at is spectra. We have mass to charge on the x-axis and intensity on the y-axis. And again, in mass spec, we're looking at a mass to charge value. So for this particular protein, this is a dimer. So it's two proteins bound together. That's a non-covalent interaction. But the mass to charge that we get here is for that dimer structure. So we can maintain these structures going from solution to the gas phase. What's shown down below is when we have the same protein bound now to a sugar. So we can see that there's 1 sugar added, 2 sugar added, 3 sugars added, etc. So we can transfer these complexes into the mass spec where we can do further characterization. I think for time, I'll move on to the last little bit. As I mentioned, one of the other things that we really care about is how these sugars can be markers for various diseases. And so we have some collaborations ongoing thinking about how these sugar-based structures change during disease progression. So the epithelial to mesenchymal transition is when epithelial cells transfer into these mesenchymal cells, which are actually metastatic tumor cells. And so we're looking at things like the glycan structures and how they change in these processes. And so this is just one biological example. But with that, what we've talked about is really that these carbohydrates have very diverse structures and functions. And so the main goal of my group is to develop new analytical tools to characterize these structures. We care both about the carbohydrate structure as well as the glycoprotein structures. So when the glycan or these sugars are also bound to the protein. And then this is some of the most recent work that we published. This actually came out last week, again, looking at how we can develop methods that enable us to look at different sugar structures. We can get signals that are different, enabling us to characterize structural differences. And so this is really the main focus of the career NSF that I have. The other thing, the career fosters an education plan. And so the education plan that we're working on in my group is developing workshops for K-12 teachers. So having done this a couple times already, a lot of the science teachers have commented that it's really challenging to have workshops specifically focused on science. And so we develop like labs that can be done in the classroom in high school settings. These are then taught through the workshops, as well as we provide these materials for a variety of K-12 teachers to use. And lastly, this is pictures of my group. So up here we have the graduate students and postdocs in my group. And this is my group of undergrads. They all couldn't come when we were taking this picture. So they took a picture at a conference that they all went to last fall. Two of them have since graduated, Emily and Amy. So we haven't taken a new picture this year because of COVID. And so with that, if there's any questions, I'm happy to answer them. It's very weird on Zoom because everybody is so silent.
I have a question, Dr. Gallagher. What topics is your lab preparing for the teaching materials?
So for the teaching materials, it's really pretty broad. We do focus on topics related to the research in our area. So that could be analytical methods. So how can we characterize molecules? That can be biomolecules in general. The last workshop that we did focused on this idea between structure and function and how that's related. So there are certain enzymes that only recognize certain sugar-based structures. So our body has developed these molecules that are highly specific for other molecules. And so the topics in the workshops are pretty broad, but they cover those areas pretty well. Are there any limitations to using the current machines we have for characterization? Yes. So one of the challenges with glycans, we go back up to what the structure of a glycan looks like. A lot of biomolecules are characterized in general based on how they interact with light. So for instance, if you're trying to determine the concentration of your protein that you have, we often take a UV vis measurement to see how it absorbs light. Now, to absorb light, most molecules need to have double bonds. In glycans, we don't have double bonds, and so they don't interact with light very readily, which makes them hard to characterize by kind of these traditional tools. Even within mass spec, one of the things that I mentioned was that we measured the mass of molecules, or of ions in particular. But these two molecules have the same mass, but one has a hydroxyl that points this way, and one has a hydroxyl that points this way. So this is something that even with mass spec, we cannot very carefully distinguish because again, what the instrument gives us is just mass. And so we have ways that we are working on that we can get more information about structure. So for instance, we can take a glycan and break it into pieces. That can give us some information about where, like particularly if we break this across the ring, we can get more information about how these sugars are bound together. We can get information about the monomers that are attached if we break between the rings. We can also use separation techniques to try to get more information about some of these very small structural differences. And part of the methods that we are developing are targeting being able to distinguish these very small structural differences using mass spec, coupled to other techniques. Does that answer your question? Thanks. So does my research focus on synthesizing glycoproteins, or is it just focused on the chemical structure function? We do have a project. So all of the questions that we're trying to answer are really related to the structure function relationship. However, we do have a project where we are working on expressing glycoproteins so that we can look more closely at how specific changes in the glycan affect the function of the protein, or the glycoprotein as it is. Does that answer your question, Bryce? Great. Anything else?
All right, well, if there aren't any further questions, thank you so much, Dr. Gallagher, for being here and talking about your research. We really appreciated it.
You're welcome.
Awesome. I will go ahead and share my screen with the sign out so everyone can get their points. All right, you guys, thank you so much for attending. I hope you enjoyed listening to our 3 NSF award winners. Just go ahead and fill out that form on the QR code so you can get your point, and then stick around for our post-meeting social. It'll be on Zoom. We'll have a lot of fun, so just stay around for that. And then also, the officers and I are still in the sub. We'll be here until 8:30. So come by, say hi, and pick up your burst shirt. You can see Sean's wearing it if you forgot what it looks like.